Hello everybody, and this video will be about control of microbial growth, physical methods and chemicals. It is clear about that it is necessary to control the microbial growth, to not allow them to make an overgrowth, and there are two basic ways to prevent overgrowth of bacteria of microorganisms by killing microorganisms or by inhibiting the growth of microorganisms. Control of growth usually involves the use of physical or chemical agents, which either kill or prevent the growth of microorganisms. So, if this agent kill the cell, we call them cidal agent. If the agent prevent the overgrowth, we call them static agents. For example, bactericidal, bacteriostatic, and like that. Okay, let's start with physical methods. Physical control includes such methods of control as the high and low temperature, desiccation, osmotic pressure, radiation, and filtration. Sterilization. Sterilization is a process when we can kill all microbes in some object. So it's a complete destruction of or elimination of all viable organisms. It's a 100% elimination. So after sterilization, no one microorganism must present in some object. And there are no degrees of sterilization. An object is either sterile or not. Object sterile means when there are no one microorganism in this object. And by sterilization, we can use temperature, heating, radiation, or some chemicals, or physical removal of the cell. One of the most important method of sterilization is sterilization by heat, and it is widely used. For sterilization, we consider type of heat, time of application, and temperature to ensure destruction of all microorganisms. And the most resistant to the heating is the endospores. That say that it is necessary to use heat during necessary time and it take more time to kill endospores and than it need for kill other microorganisms, other form of microorganisms. Okay, there are a few methods of sterilization by heat. Insertion, so to kill bacteria by the fire, in the fire, as you can do when you try to sterilize the loop. Also, boiling, it's a way of sterilization when you boil some liquid and the temperature is 100 degree and the time of sterilization must be about 30 minutes to kill all microorganisms in that object, because you know that endospores they more stable, more resistant for heating. Also, we have such method as autoclaving, and in autoclaving we use steam under pressure, and the temperature is about 121 degrees, and pressure is about 150, and time of sterilization is not less than 50 minutes. And this method is useful for sterilization anything that heat stable, and any object that heat stable. Uh, but heat label substance will be denatured or destroyed. That must you must know about it because if you use some materials which are not heat stable, you should not sterilize it by autoclaving. Also, dry heat, hot air oven. Um, and it just that sterilization by heat with 160 degrees during 2 hours, and usually this method can be applied for glassware, metal, and objects that won't melt. And this in this page, you can see autoclaving. This device is called autoclave, and there are two chambers. One chamber is for object, and second chamber invisible on this page. Second chamber is for water. In the process of autoclaving, water must be put into the second chamber, and uh, and this water became boil, so like boil, and then a little hole must be opened. This little hole connect the second chamber and first chamber, and steam from the second chamber will enter to the first chamber, and pressure will be created, and high temperature and steam, and this is what will kill bacteria, what will kill all microorganisms, and including the endospores. And this is very important, but because not all measures can destroy endospores, but by autoclaving you can destroy endospores. And but it must be noted that in autoclaving you can't sterilize heat label object because they will be destroyed. Oh, and also in this page, you can see how does it work. So this is second chamber and this is first chamber. In first chamber, you put the objects for sterilization, and in the second chamber, you put water, and water became to boil. And after opening the hole, the steam with the pressure will enter to the first chamber. Dry heat, dry heat. Okay, in dry heat we use specific this device. It's a metallic chamber made of steel or aluminum, and it has a inner chamber when you should put your objects, and it is very close to oven. Oven it's a device which you use in your kitchen to cook some cakes, because you have some device made by steel, oven made by steel, and you put some cakes here, and the temperature will be more than 100. And this device is very similar because it works like a oven. It just keep high temperature, uh, and this higher temperature, higher air with higher temperature. So this is a main reason. This is a main thing that we use in this process. Next, it we just talked about sterilization by heat. And so next method is sterilization by irradiation. Irradiation usually destroy or discharges nucleic acid. Ultraviolet light is usually commonly used to sterilize the surface of object. Also, X-rays and microwaves are possibly useful. And maybe you have saw in some hospitals they use ultraviolets to sterilize object, and of course they sterilize the air by this way. Next method, it's a physical removal and filtration. And by specific filters, you can sterilize some solution, because these filters they enable, because some bacteria and eukarya they can't go through these filters, but viruses they can. This method is not available for viruses, because viruses are too small, and that's why we can use this method only for sterilization, only for killing bacteria, only for removing eliminated bacteria and eukarya. Chemicals and gas, toxic chemicals kill a forms of life and specialized gas chamber. And also pasteurization. Pasteurization, it's not a kind of sterilization, because it's a mild heat to reduce the number of microorganisms in a product or food. So sterilization kill all microorganisms, remove all microorganisms, and sterilized object must not have any microorganisms. But by pasteurization, you can reduce the number of microorganisms in the object. Uh, in the case of sterilization of meals, the time and temperature depend on killing potential pathogens that are transmitted in milk. For pasteurization of milk, you should use 63 degrees for 30 minutes, and then flash method 71 degrees for 15 seconds. Physical agent to control microbial growth, low temperature. We usually put in refrigeration some product uh, to keep this product, this food in good condition. Because you know that bacteria they make some harm in food. But if you put this product in the refrigerator, you can stop the multiplication of microorganisms, and you can prevent the harmful activity of the microbes. But not to kill bacteria, just stop but not to allow them multiplicate. And also drying, removal of the water. Most microorganisms are sensitive for drying, so they can't grow without water. They need water to multiply and grow. And so by drying, you can you can stop, you can prevent the overgrowth of microorganisms. Next question is the chemical agents. Chemical agent can kill or also stop bacterial overgrowth. Okay, by chemical agents, we can make a disinfection. Disinfection is the elimination or inhibition of pathogenic microorganisms, in spite of sterilization when we kill all microorganisms. But by disinfection, just eliminate only pathogenic microorganisms. As a disinfectant is chemical agent used to disinfect inanimate objects such as a work surface and floors. So we can't use disinfection for the animals and people, only for the some surface. Disinfectants are incapable of killing spores within a reasonable time period and are generally effective agents and a narrow range of organisms that physical needs. Types of disinfection: air disinfectant, alcohol, phenol, and oxidizing agent by its chemical structure. Okay. Disinfectants, phenols, uh, oxidizers, alkalizing agents, and some others like sulfides and let's say. Classes of disinfection: soaps and detergents, acids and alkalis, heavy metals, halogens, alcohols, and all these disinfectants can be used for disinfection, but they have different way of activity, different mode of action, and different spectrum of activity. And next question is antibiotics. Very important question. Okay, here you can read history of discovery antibiotics. Alexander Fleming was the first who opened antibiotics, and he discovered that molds can prevent the growth of Staphylococcus. And then in 1940s years, the first antibiotic penicillin was created. And in Russia, this discovery was made by Zinaida Vissarionovna Yermoleva. And what is antibiotics? Antibacterial drugs represents a chemical substance derived from biological source or produced by chemical synthesis that is able to destroy or inhibit the development of growth of bacteria. Classification of antimicrobial drugs. So it can be classified by target, pathogen, so it can be active against bacteria, against viruses, against fungi. By the source of antibiotic, they can be natural, semi-synthetic, or synthetic. Natural is mainly fungal sources. They can be taken from some fungi like penicillinum, like cephalosporinum, and also from some bacteria like actinomyces. They also can produce antibiotics. Also like Bacteroides, Pseudomonads, they also produce some antibiotics. And also as a natural source, it as a natural source, some plant can be, and also some animals, for example, uh, in from the animals we can take lysocin, which kill bacteria. But it is one problem with a natural antibiotic because they are very spread in the nature, and you know that bacteria they can mutate very quickly. Uh, they are ready to change to for they are ready to change themselves to be to be resistant to the antibiotics. That's why if they know if bacteria know the mechanism, the mode of action of antibiotics, they will break it. And if antibiotics is very spread in the nature, in the human population, this bacteria will not be sensitive for this antibiotic because they know this mode of action and they can overbreak it. That's why natural antibiotics usually are not effective. And semi-synthetic, they are made by chemical modification of natural antibiotic, and synthetic, they are wholly synthesized in the laboratories by biochemical synthesis. And the synthetic drugs, they have an advantage that the bacteria are not exposed to compounds until they are released. They are prepared to exhibit greater effectiveness and less toxicity. And on this slide, you can see the dependence between toxicity and effectiveness. Natural antibiotics, they are higher toxicity and less, and have a higher toxicity and less effectiveness. And synthetics, they are the best because the effectiveness, uh, they toxicity is not so high. Okay, next classification is based on how do they work. So kill them bacteria or just stops their growth. So they can be sidal or static. And the bispectrum of activity, they can be broad spectrum or narrow spectrum. Broad spectrum kill all bacteria, most of bacteria, but narrow spectrum acts against specific family of bacteria. Only one specific group. Broad spectrum antibiotics, they can be used in the case if you don't know the etiology, if it is not clear which actually bacteria cause this infection, but this patient need the treatment. But disadvantage of this type of antibiotics that that they also kill the normal microbiota, bacteria from normal microbiota. But narrow spectrum, they can be used in the case if you actually know the etiology, you know which group of bacteria cause this disease, or the genus or species of etiological agent. And so in this case, you can use narrow spectrum. Narrow spectrum is the best because it does not kill normal biota. Classification of antibiotic drugs on chemical structure. So we have a few classes: beta-lactams, macrolides, fluoroquinolones, tetracyclines, aminoglycosides, and others. And the main classification is based on the targets of antibiotics, and targets of antibiotic must be selective. So it means that it must be each mode of action must have one target, and this target must not be in the eukaryotic cell. It must, because it will be toxic. It will have high toxicity if the target is not unique. Okay, and by target site, all antibiotics are divided into antibiotics that destroy the cell wall, antibiotics that destroy the cell membrane, antibiotics that stop the protein synthesis, antibiotics that stop the synthesis of nucleic acid, RNA and DNA, and antimetabolites that destroy the metabolism of bacteria. So cell wall and killing antibiotic, cell wall destroying antibiotics and beta-lactams, glycopeptides, and bacitracin. And on the plasma membrane acts polymyxin, lipopeptide, and to the protein synthesis, we can to the protein synthesis act such antibiotic as aminoglycosides that recyclize, they connect with a small subunit of ribosome and stop the protein synthesis. And with big subunit of ribosome, interact macrolides, lincosamides, chloramphenicol, and oxazolidones. To the nucleic acid can influence such antibiotics like fluoroquinolones, and they destroy DNA synthesis, and on RNA synthesis influence rifampicin. And antibiotics that influence on the metabolic pathway, it's a folic acid synthesis, like sulfonylamides, and mucolic acid synthesis, like isoniazid do. So here you could say also classification by mode of action that we discussed previously, and let's talk about each mode of action. One of the big group of the big group of antibiotics are influence on the cell wall biosynthesis, and the target is peptidoglycan, because this is the basic substance of each cell wall. And antibiotics that destroy cell wall, they usually act as a bactericidal agent, so they kill bacteria. And also they have selective toxicity because eukaryotic cells, they does not have any peptidoglycan. So which antibiotics act influence on the cell wall? Of course, penicillin, which was the first antibiotic discovered, is one of the several antibacterials within a class called beta-lactams. This group is compounds includes penicillins, cephalosporins, monobactams, and carbapenems, and is characterized by the presence of B-lactam found with the central structure of the drug molecule. The beta-lactam antibiotic blocks the cross-links of peptide chains during the biosynthesis of new peptidoglycan in the bacterial cell wall. They are able to block this process because the beta-lactam structure is similar to the structure of the peptidoglycan subunit composed that is recognized by the cross-linking transpeptidase enzyme, also known as penicillin-binding protein, PBP. So look at these slides. Here you can see different beta-lactams antibiotics, and this is beta-lactam rings. You can see that each of these antibiotics has beta-lactam rings. And how does they kill bacteria? They influence on the small enzyme which is called transpeptidase. Transpeptidase is necessary to connect peptidal chains. This is N-acetylglucosamine and acetyluramic acid. This is a part of peptidoglycan, and this is peptide chains. Maybe you remember from the first lecture that peptide chains, it's also part of peptidoglycan. And transpeptidase, it's a small enzyme which is necessary for connected to this part. But penicillin, it can interact with this transpeptidase and stop the process of connecting of the parts of peptidoglycan. So destroy peptide change. Okay. Cephalosporins, it's a modern antibiotics which also belong to the group of beta-lactam antibiotics, and Also, they block the transcriptase activity. And However, beta-lactam ring of cephalosporins is fused to a six-member ring rather than the five-member ring found in penicillin. So its structure different than the structure of penicillins. This chemical difference provides cephalosporins with an increased resistance to enzymatic inactivation of beta-lactamase because uh, penicillins they can be destroyed, but specific enzymes which are produced by bacteria, and this enzyme is called beta-lactamase. Cephalosporins also was isolated from the fungus, so this is a natural antibiotics, and family of semi-synthetic cephalosporins is much larger than the penicillins, and these drugs have been classified into generation based primarily on their spectrum of activity, increasing in spectrum from the narrow spectrum. A new fifth generation cephalosporins has been developed that is active against methicillin-resistant Staphylococcus aureus, which is not sensitive for the other groups of beta lactams. Okay, here we can see spectrum of activity, and they have different spectrum of activity, and some semi-synthetic are broad spectrum, as for example third and fourth generation, but the first generation is narrow spectrum of activity, very closely to the penicillin spectrum of activity, things like that. Next mode of action is inhibition of protein biosynthesis, and to stop biosynthesis of the protein, but antibiotics interact with the ribosome, small unit or big unit. Okay, major classes of protein synthesis inhibition antibacterials. Chloramphenicol, macrolides, and glycosamides. They bind to the 50 to the small ribosomal subunit, which have 50 sedimentation, and prevent peptide bond formation, stop protein synthesis. Look at this slide. So this is a large subunit, and large subunit, and this is a antibiotics which destroy the process of protein synthesis. Just stop the protein synthesis by blocking the large subunit. Aminoglycosides bind to the 30% sedimentation ribosomal subunit and small subunit, and also impair proofreading, resulting in production of faulty proteins. And tetracyclines also bind to the small subunits and block the binding of tRNAs, thereby inhibit the protein synthesis. Okay, so antibiotic which interacts with the small subunit, they can block association with RNAs with ribosome. They destroy the process of tRNA connecting with ribosome, and also they can cause a mismatch between colons and anticodons, leading to faulty proteins that insert into a disrupt cytoplasmic membrane. So they make the process of protein synthesis to be wrong. So protein will be continue to synthesize, but the protein will be wrong. They will be unworking. And when this protein will became the part of cytoplasmic membrane, the cytoplasmic membrane will be destroyed. So this is a mechanism of bactericidal effect. But if this antibiotic just block the association with DNA, RNA, and ribosome, this its effect, its action will be like bacteriostatic. If antibiotics blocks large subunit, big subunit, um, it also destroys the process of protein synthesis, so blocked peptide bond formation between amino acids, and also its action is like bacteriostatic, not to prevent the overgrowth, stop the multiplication. Okay, here you can see how aminoglycosides act. Tetracyclines, um, they blocks the small unit mRNA connection with the small units, and chloramphenicol, it blocks big unit. Next, antibiotics. It's antibiotics which inhibit the membrane function. It's a small group of antibacterial targets the bacterial membrane as a mode of action. The polymyxins are natural polypeptide antibiotics that were first discovered as a product of Bacillus polymyxa. Only polymyxin and B and polymyxin E have been used clinically. They are lipophilic with detergent-like properties and interact with the lipopolysaccharide component of outer membrane of gram-negative bacteria, ultimately disrupting both their outer and inner membranes and killing the bacteria cell. Unfortunately, the membrane-targeted mechanism is not selective toxicity, and these drugs also target and damage the membrane of the cell in the kidney and nervous system when administered systemically. That's why it is necessary to not administer this drug systemically, only local. Okay. So the target of polymyxin, it's a lipopolysaccharide, some lipids in the inner member of gram-negative bacteria. Next, it's antibiotics which stop the process of nucleic acid synthesis. And to this group, we to this group belong such antibiotics like metronidazole, rifampicin, and anti-tuberculosis antibiotics. Metronidazole is a semi-synthetic member of metronidazole family that is also antiprotozoan. It interferes with DNA replication in target cell. And so the target of metronidazole, it's a DNA gyrase. It's an enzyme which make a DNA like a spiral form, make a DNA in a spiral form, and it is necessary for process of multiplication. So metronidazole stop bacterial multiplication. And the drug rifampicin is also semi-synthetic, and it is belong to the family rifampicin, and it can block the RNA polymerase activity in bacteria. By this way, it's it's block the process of RNA replication, polymerization of RNA, and this antibiotics, it has a selective toxicity against bacterial cell. and it can be usually administered in the case of mycobacteria infection. Despite the selectivity of its mechanism, rifampin can induce liver enzyme to increase metabolism of other drugs being administered and leading to hepatotoxicity and negatively influence the broad viability, the therapeutic effect of the common drugs. One member of the quinolone family, a group of synthetic antimicrobials, is the nalidixic acid. Nalidixic acid selectively inhibits the activity of bacterial DNA gerase, blocking DNA replication. Chemical modification to the origin quinolone backbone have resulted in the production of fluoroquinolones like ciprofloxacin and levofloxacin, which also inhibit the activity of DNA gerase. The spectrum of activity of this antibiotic is broad spectrum against gram-positive and gram-negative bacteria, and they act like a bacteriostatic antibiotics. So they block the process of bacterial multiplication and growing. Mm. And the last one group, it's the antibiotics which inhibit the metabolic pathway. Antimetabolites, they also can be called. And one of the big group from which acts with this mode of action, it's a sulfanilamide, sulfa drugs. They are the oldest synthetic antibacterial agent, and the target of these antibiotics is folic acid. So they interact with bacteria by concurrence activity, and they take all folic acid, and, and bacteria can't and bacteria have no any folic acid. Folic acid is a acid which is necessary uh, for growing and multiplication. So without folic acid, bacteria can't multiply. Uh, that's why this mode of action is have a bacteriostatic effect. And And so we just discussed the types of antibiotics, and we have about 600 antibiotics which we can use in our medical practice. But now we have one big problem, and the name of this problem is antimicrobial resistance, because bacteria, because microbes, they are very changeable organisms. So they can change their genome, they can change their behavior, they can change the sensitivity to the drugs. And this process became very quickly. That's why we have so many antibiotics, but not of them can work. Um, and bacteria, because bacteria can overcome antimicrobial compounds. Uh, uh, Several important factors can accelerate the evolution of drug resistance. This includes overuse and misuse of antimicrobials, inappropriate use of antimicrobials, subtherapeutic dosing, and patient non-completions with the recommended course of treatment. In a few words, that if some patient stop the therapy with antibiotics uh, before deadline or before it must be completed, uh, he makes bacteria to be resistant. Using lower doses, short courses of antibiotics make bacteria to be resistant. So we have two types of resistance. Origin of drugs resistance is classified in non-genetic origin and genetic origin. Genetic origin may be encoded on bacterial plasmid and chromosomes mutation. Drug resistance are found in plasmids or in transposons that can be transferred easily between microbes through horizontal gene transfer. Non-genetic drug resistance microorganisms that are non-multiplying may be phenotypically resistant to drugs, persistent mycobacteria. Microorganisms can lose the specific target for an agent for several generations and thus be resistant. Microorganisms can appear in the host sites where antimicrobials are excluded or not active. Mechanism of resistance to the antimicrobial agents. How bacteria can overcome the action of antibiotics. One of the mechanisms is modification, destroying, and activation of antibiotics. One of the best example is the production of beta-lactamase. Beta-lactamase is an enzyme which can destroy beta-lactam ring of beta-lactam antibiotics. Some bacteria can produce this enzyme and destroy by this enzyme the antibiotic. Second, this is uh, when microorganisms decrease their permeability, such that effective intracellular concentration of the drug cannot be achieved. So they antibiotics doors the doors will close and antibiotics can't enter. Um, so next mechanism of resistance is changing the targets. So if the target will be changed, the antibiotic can't interact with it. Next, it's a multi-drug resistant pump. Efflux mechanism means that when antibiotics enter to the bacterial cell, it is um, very quickly will be eliminated from the bacterial cell. and this is the changing of metabolic pathway. For example, when some antibiotics, for example, sulfonylureamides, they take all folic acid and bacteria can't multiply because it need folic acid for multiplication. Bacteria can change its metabolism and exclude folic acid from its metabolism, change metabolic pathway, and continue to multiply without folic acid. So here you can see all of this process. Inactivated by acid to destroy antibiotic by the acid. Decreased uptake. So antibiotics can't enter to the bacterial cell. Also, to efflux pump sound when antibiotic came to the entered to the bacterial cell, but very quickly eliminated from the bacterial cell without action, and changing the target. When target is changed, antibiotic can interact with it. To choose the correct antibiotic for the therapy, it is necessary to know the sensitivity of bacteria, and how we can find this information by specific antibiotic sensitivity test. It can be diffusion test or dilution test. Diffusion is qualitative, dilution is quantitative. Diffusion test just answer all the questions which antibiotic will be effective, but dilution test also will give the information about the concentration of antibiotic which will be the best. So dilution test, one of the dilution tests, it's a broad dilution test when the some antibiotic must be diluted from bigger concentration to the lower concentration, and then bacteria will put in each of these tube, and then the stage of incubation, and after incubation we should examine all this tube and find the tubes where we don't have any visible growth. And this is clear, this is effective concentration. But from this tube, we can see that growth of bacteria began to appear. And this means that this concentration does not affect it. So this is a maximal, this is a minimal concentration which will inhibit the growth. MIC, minimal inhibition concentration of antibiotic. So minimal concentration of antibiotic which inhibit the bacterial growth. And this diffusion method, it's a very spreaded method, a widely spread method, which we usually use in laboratory to check the sensitivity, antibacterial sensitivity of the pure culture. So how it can be done? Test organisms must be put to the nutrient media by the streaking method, and then paper which consists of different antibiotics also must be put to the media, and then incubation. And if bacteria is sensitive to these antibiotics, it will not grow nearby this antibiotic, and we can measure the zone of the growth absence. And if if there is no any zones of growth absent and bacteria can grow nearby the antibiotic, that means that this bacteria is resistant to these antibiotics. So this is suspectable. This bacteria is suspectable for this antibiotic. For this is we have intermediates suspective sensitivity, and for these two antibiotics, this test of microorganisms is not sensitive because it is resistant. And here you can see the result. On this plate, you can see that all antibiotics are effective and bacteria can't grow nearby this antibiotic. But here we can see resistant strain. It can grow nearby 1, 2, 3, 4 antibiotics. Only 2 antibiotics can prevent the growth. So only 2 antibiotics can have a good effective, and one of antibiotic is have a middle intermediate effect. Okay, and zones of inhibition of microbial growth around the disc are measured and compare with the norms, and by this way you can decide if the antibiotic is effective or not. And also, like previously method, we can use e-test. It is very similar. So this paper consists of one type of antibiotic by in the different concentration, and from in this method, it is necessary to test microorganisms to the nutrient media, then put the paper with antibiotics with antibiotic with different concentration. And if antibiotic will act, the bacteria can't will not can the bacteria will not grow nearby these antibiotics. And the minimal concentration which prevent the growth uh, will be measured. And this is a minimal inhibitory concentration of antibiotic, minimal concentration which prevent the growth of bacteria. Thank you for your attention. If you have any question, please ask me in commentary.